welcome to another episode, the second part of the introduction of the Bioinformatics Beginner Course, where we'll be discussing about the accessing databases of bioinformatics. That is the tools that you'll be exploring to perform your bioinformatics work. So now we'll be discussing about the databases, and I would like to take us through some of the databases. So basically, there are two classification of databases for bioinformatic analysis. One is the nucleotide, and also the other part is the protein part, protein database. And in between, we have the specialized databases that perform specialized function. So in the nucleotide part, I would like to go through it very fast. So we have the EMBL, the European Molecular Biology Laboratory. We have the gene bank, the EMBL, so the website is ebi.ac.uk. We are still going to talk about them when we are discussing, when we are showing us how to use them. And also the gene bank, you must have come across the gene bank, the NCBI. So the NCBI is an American database for nucleotide sequences. It's a very rich resources. It's a very rich resource for any of the analysis that you want to do. We also have the DNA Data Bank of Japan, is also a very rich resource, but most of the time the Gene Bank NCBI is what most bioinformaticians to explore. Although I haven't had the course to explore what is inside DDBJ because of the resource I'm looking for. But I've heard of the which maybe during this presentation we are still going to explore it and know or see how it looks like. So now altogether, these nucleotide databases are under the body called International Nucleotide Sequence Database. And apart from this database, we have the whole genome database, which is called the Ensembler. So we are still, hopefully, we get to that point in the. No, this might not be in the introductory part, but we just have to know about them. That is that about the nucleotide database. Moving on to the protein database, there are three different classifications of protein. You have the sequence part, you have the interaction part, and you also have the structural part of a protein database. Now, under the sequence part, you have the UniProt, the PIR, the Swiss prot. And also under the interaction part, we also have the BioGrid, the string. We also have the under the structural part, we'll have the PDB, we have the cath, we have the scope. Each of these databases, they have the functions and the roles they perform. So, for example, in the sequence, so they deal more about the primary sequences, primary protein sequence that you submit. While the interaction, the protein-protein interaction, you have the biogrid and string. So each has the role they play. So in the structural part, the full meaning as can be seen in the slide is the protein data bank. The cath is the class architecture topology homology, and we also have the structural classification of protein. Now in between these two, there are specialized databases that one of our presenters in the second webinar we had, Austin, talked about the gene expression omnibus and how it can be used. This is not for the beginner class, so but just have an idea of what these databases, what they do, what is this all about, and how they fit into the category of databases for bioinformatic analysis. Now, when you search for databases, you can see something of such like this, whereby different databases. There are hundreds, there are loads and loads of databases online, as we are going to be showing us. For example, let us take us through this particular one, whereby when we search for bioinformatic databases, you can see a lot of databases online. So now, as a beginner, you come to realize that it's not all these databases that you'll be needing. It's not all these databases you'll be using for your work, for your analysis, or to gain more knowledge about to give meaning to your biological data. So now, if for example, let us go back to the slides that we are showing. Now, as a genomic expert, so you might not have anything to do. You might, although you still have a contact with all these protein databases, but as a genomics from the beginner level, you'll have to deal majorly with these nucleotide databases. So those are your forte. But when you're talking about those that are into proteomics, transcriptomics, or epigenetics, that's when you'll be dealing with all these protein databases, and then the gene expression omnibus, or the OMIM, the inherited disease database, which is the online Mendelian inheritance of man. So these are the databases in short, and how we are going to use them, how we are going to be using them will be what we'll discuss in the next part of this introductory session. See you in the next presentation.